Welcome to MolBiolect. My name is Ankit and today I am going to talk about the human monoclonal antibodies that are going to work against the SARS-CoV-2 infection. So before starting on with the video, please like, share, comment and subscribe. So let's get into the video. So the severe acute respiratory syndrome coronavirus 2, or the full form for SARS-CoV-2, is the agent for coronavirus induced disease 19 or the COVID-19. It emerged in China late 2019 and caused a pandemic. SARS-CoV-2 belongs to the Sabecovirus subgenus, the genus being beta coronavirus and the family being Coronaviridae. Like SARS-CoV, this virus crossed species barriers from animal reservoirs and it currently causes a fatal respiratory syndrome in humans and in some cases cats. So now how we eradicate this virus? So before talking to that, please like, share and subscribe this channel for obtaining more precise knowledge on SARS-CoV-2 infection and biochemistry genetics as taught in our advanced study courses. So let's start with our video. So how these monoclonal antibodies are prepared? Coronavirus spike proteins, okay, so these proteins were expressed in HEK293 T cells or the HEK293 T cells with a C-terminal trimerization. Okay, we must ignore the technical terms right now. So basically spike proteins reservoir was made for testing, and this was made on HEK293 cells for getting purified spike proteins. Next step, a mouse, particularly H2L2 mouse, were immunized with spike protein ectodomains from HCOV, OC43 SARS-CoV-2, SARS-CoV-2 and MERS-CoV-2. Okay, like as I have written over here, so these were made. And after 2 weeks of immunization, as I've shown over here, 2 weeks of immunization, they were killed. They were killed and they were actually hybridized with myeloma cells. So by hybridized, I mean that the spleen cells over here, these were hybridized with the myeloma cells for the continuous production of purified antibody from the mouse. And this served as a source of purified antibodies from mouse, and this was made the control for the experiment that we are talking about. If you want to know the dosage of immunization, it was specifically 25 microgram per ml for the control mouse as I was talking over here. So next, for the human monoclonal antibody, which is typically named over here 47D11, the cDNA which encodes the 47D11 H2L2 MAB. H2L2 was a mouse, okay, the mouse which was used for this model. H2L2 MAB variable regions of the heavy and light chains, and they were cloned into plasmids which contained IgG1 heavy chain and Ig kappa light chain. These are constant regions, by the way, okay, and these plasmids also have a interleukin 2 signaling system. So whenever interleukin 2 is induced, they produced purified recombinant antibodies which is named as 47D11 as isolated from humans. Okay, so the transfection of this cell with the IgG1 heavy and light chain expression plasmids, according to protocol they studied, the human recombinant antibodies were formed and they were ultimately purified. So how 47D11 actually works? The SARS virus 2, the SARS-CoV-2 actually, S1B receptor binding domain. So basically it has a S1B receptor binding domain which consists of a receptor binding subdomain which contains residues of amino acid which is nearly about 438 to 498 residues long, which loops out from the antiparallel beta sheet code domain. We don't need that basically, it's a structural insights, but this part directly engages with the ACE2 receptor. I have already explained this in my previous videos also, that there is a particular receptor binding domain and subdomain which interacts with the human ACE2 receptor, not the whole of the spike protein, you must remember this. But this S1B receptor which directly binds to the ACE2 receptor in different viruses can differ substantially. For example, in SARS-CoV and SARS-CoV-2 they differ substantially, but neutralizing antibodies are very much specific. So antibodies, which are specific for, suppose if these two are two receptors, suppose these two figures over here are two different receptors. So if one antibody is effective towards this receptor of the, not this receptor actually, spike of the virus, then the other will be similarly effective towards the other, this one which I am pointing at this virus. But it's not so. what happens is that 47D11, the one I am talking about, this has a cross-reactive nature. So this antibody can neutralize any virus of this family as far as the studies go on. So 47D11, it binds to a conserved epitope. Suppose if this is our spike protein in general, this spike protein may contain certain portions which directly bind to the ACE2 receptor. Suppose this is the ACE2 receptor I am talking about. So this part of the ACE2 receptor directly binds to this part of the spike protein. Now 47D11 binds to a conserved epitope on the spike receptor binding domain, that is in this part. Thus its ability to neutralize both SARS and SARS-CoV-2 is basically obtained. How? Because SARS-CoV and SARS-CoV-2 both contains this thing in common. Okay, this receptor binding domain is almost same, but the mechanism is independent of receptor binding inhibition. So the mechanism of how it takes place is not known yet. Also, in this experiment, they have proved that using a trypsin based switch model. So basically using a trypsin based switch model, which is nothing but if you put trypsin into the system, it actually functions or switches something on. So basically in a trypsin based switch model, 47D11 shows to impair the SARS-CoV-2 spike protein mediated syncytia formation, although a clear-cut mechanism is not. So what is syncytia formation? Basically, if I tell you in much simpler words, basically this fusion of the virus as it enters binds to ACE2 and it enters in the cell in endosome form, and then it binds to the lysosome and then forms endolysosome. So this entire, in simple term, this entire thing is blocked by the 47D11 by using some undiscovered mechanism, but other studies back up that spike may have been inactivated using antibody induced destabilization or prefusion structure as reported in other antibodies. That is nothing but basically other studies that back this up actually is that although no clear-cut mechanisms have been till date discovered, but studies say that they may have been inactivated using antibody induced destabilization. So destabilization of this structure I am talking about over here, there is a prefusion structure. So this structure over here is known as a prefusion structure, and if there is a destabilization in this part, then it will not completely bind over here. And if it does not completely bind to this part, my marker is getting low on ink, does not bind to this part, then the whole system will collapse. 47D11 may be useful in treating with other neutralizing antibodies from SARS-CoV, that is this one over here. This was found during the SARS-CoV outbreak. So this antibody's name is CR3022. So they may be utilized using along with the CR3022 antibody that shows a cross reactivity to also SARS-CoV-2. Okay, so I would like to conclude my video now. So in conclusion, what I can say from this entire mechanisms as I have made you understand is that SARS-CoV-2 is neutralized by 47D11, which is a first report of human monoclonal antibodies serving to neutralize SARS-CoV-2. So if you like this video, also first of all, thank you guys for helping me complete 200 subscribers. And also if you like this knowledge boost, and then please like, share and do subscribe to my channel. You don't know that your subscription gives me so much motivation to work on with my next videos. And also never miss an update by tapping that bell icon on tapping that bell icon on my channel, so that you will never miss an update on education or cutting edge research simplified by me. Okay, so for now, Moy Biolect is out. Please stay at home, stay safe, and also don't panic, and also keep yourself protected from the outside world right now. Okay, stay healthy, stay safe, stay at home. Bye.